Welcome to State of the Science. This is a new way of talking about research in the field of type 1 diabetes. We have a global audience of scientists here today. We thank everybody for joining, and I'll briefly introduce our panelists. Two panelists will sort of set the stage for the discussion, and then we'll ask them to weigh in on their thoughts for 3 to 4 pressing questions in the arena. Questions are sort of free-flowing, like a cross-idea exchange, town hall, and a respectful debate. We'll pause at 3 different time points to conduct polls that were sort of selected by the panelists, and we'll have the panel weigh in on the audience voting and feedback. And I'd like to take a moment to recognize our fantastic sponsors, 10x Genomics, Vertex, Prevention Bio, Imsice, and Inspiro. These organizations are at the forefront of cutting edge science, and without their support, we couldn't really bring you the state of the science. So you can find more about their innovative companies on our website and at our Ask the Expert series, both live and archived on our YouTube channel. So a few pro tip for attendees: if you want to submit a question, place it in the Q&A, and you can also submit questions throughout the presentations. Participants can upvote favorite questions, and at the end of the session, we'll answer questions as time allows. Event is to be recorded live and on the Sugar Science YouTube channel, and will be housed there for further viewing. So in the interest of saving time for our discussion, I'm going to do some brief introductions for each of our scientists, and you can connect directly with them on our website in the collab, where the majority of our members are, hanging out, or reach out directly. And I just wanted to have Mark Atkinson from University of Florida give a few words.
So thanks, Monica, and thanks to everybody for joining today. So I've been a type 1 diabetes researcher for about 38 years this August. And I'll say that the topic of today's talk about mitochondrial dysfunction, ER, stress, and pro-hormone processing has clearly been an orphan research, and that's not meant in disrespect for today's speakers. I think that this is one of the most exciting areas of type 1 diabetes research right now. For too long, it was relatively relegated to notions of type 2 diabetes, but as time goes on, we're increasingly understanding the role of beta cells in the formation of type 1 diabetes. And I think that with the work of the group that will speak today, this area is rapidly accelerating itself to the forefront of type 1 diabetes research, because there's so many important questions about the role of beta cells in the disease. So I just joined Monica in thanking the speakers for participating, and look forward to some really good discussions. So that's my intro. I'm excited.
That's great. Okay, yeah, it is a really interesting, cornerstone of type 1 diabetes research, and I can't wait to hear what everyone has to say. So I'm just going to introduce people sort of just in one by one. Amelia Linneman Phd, she's from Indiana University. Yeah, you can do a little wave, that's great. Assistant Professor of Pediatrics. She has she's a Hearn New Investigator Award recipient. She has a KO1 grant. She's an NIH NIDDK Gateway Investigator recipient. And the primary focus of her lab is to understand mechanisms of pancreatic beta cell death and survival, islet compensatory adaptation to cellular stress, including autophagy, and how these factors contribute to diabetes pathogenesis. Scott Soleimanpour is an MD from University of Michigan. He's an associate professor at the Department of Internal Medicine and the associate director of the Elizabeth Weiser Caswell Diabetes Institute. His lab is primarily focused on the control of pancreatic islet beta cell function in diabetes by all aspects of the mitochondrial life cycle: biogenesis, dynamics, and mitophagy. Okay, I'll skip over that. Peter Thompson is up in Canada at the University of Manitoba, and he is an assistant professor in the Department of Physiology and Pathophysiology at the University of Manitoba, focused on the role of beta cell stress responses in T1D, as well as the mechanism of secreted EVs, extracellular vesicles in T1D. Bridget Wagner, PhD, is over in Boston at MIT The Broad, and she's the director of pancreatic cell biology and metabolic disease in the Chemical Biology and Therapeutic Sciences program at the Broad Institute of MIT and Harvard, where she's also an institute scientist. And her group's research really focuses on the chemical biology of diabetes, with the aim of identifying small molecules capable of increasing pancreatic beta cell number and function with the ultimate goal of discovering new therapeutic approaches for diabetes. And finally, Mark Atkinson, University of Florida, MD. He's a giant in the field of type 1 diabetes, and his research is broad in scope, but directed at identifying prevention or cure for type 1. So thank you again for everyone joining us. You are all well known in the field and esteemed, and we appreciate your time. So Dr. Linneman and Soleimanpour, would you like to set the stage with your impression of where things stand in relation to mitochondrial and pro-hormone dysfunction in beta cells?
Scott, you want me to start or you want to start?
I will defer to you.
All right. So, I think that I'm a little biased in terms of thinking about autophagy because it's something that my lab studies and we're really interested in. But, thinking more broadly in terms of how that fits in with mitochondrial dysfunction and pro-hormone processing, I think that in general in the field, there is growing acceptance that the beta cell plays, this key role in its own demise in diabetes, especially in type 1 diabetes. And the question is, do some of these defects that we see occur prior to diabetes onset or as a consequence of, hyperglycemia and some of these other factors that are playing a role in the process. So I think that something I'm very interested in is how to differentiate between cause and consequence. There are certainly clues in terms of type 1 susceptibility genes. There's this whole other set of type 2 susceptibility genes as well. But what can they tell us about distinguishing between the two? So that's something that's on my mind. I won't take up too much time here. But the other thing is, thinking about how we study these processes. We know a lot, but really do we? Because many of the systems we use are contrived. And so how much of what we know is due to how we actually study it? So Scott, I'll let you kind of jump in here.
Thanks, Amelia. That was great. So first, just thank you, Monica and Mark, and then to my, colleagues here. It's really, really exciting to be doing this with all of you and to see all of you, although hopefully one of these days we'll be doing these things in person again. And for everyone tuning in as well, I appreciate it. So I think as the token physician scientist on the panel, I feel like I have a couple of responsibilities, but I'll just start by saying this. It is only relatively recently that we've appreciated the beta cell's role in type 1. I personally have a lot of battle scars from trying to take on that fight from work in my lab. For many years, people telling me I was wasting my time studying beta cells and type 1 genetics. So it is great that we are at a place now where we can be on a call as beta cell biologists and immunologists and talk together about the disease. So that's very big picture. But I think regarding the topic that you set up, I think the one way I would look at it and I think in type 1 overall now, we know now type 1 is not purely an immunologic disease. We know it's not a beta cell disease. We know it's not a microbiome disease. It's an everything disease, exocrine pancreas too, right? So things are broad. There's a lot of players involved. It's complicated. I think that very much is specific to how we think about mitochondria and pro-hormone processing as a topic in the beta cell. It is very easy to zero in on one mechanistic pathway and say, that's the one. I would love it if it was as simple as that, but we're not studying monogenic diseases. And so I think it's important to realize that a lot of these processes are linked. There are multiple causes. I'm a big believer in there's a big genetic role. And so I think that you can learn a lot from type 1 genetics, but clearly there's an environmental component too. But I think we have to not be so, we have to look at the disease broadly, I guess, as a physician, not with blinders on, but realizing that there's multiple players and these things interact and regulate each other. And while I think each of us has a topic of interest, obviously I'm very focused on mitochondria. That's what my lab studies. I also recognize it's not as simple as that, right? So, things that are going on in Peter's lab and Amelia's lab and Bridget's lab and Mark's lab, it's all working together to put these puzzle pieces together and realize that. So anyways, I think that's just how I will start it off, and just say that I'm excited about hearing what my colleagues have to say, because I think we come at it at different angles, and that's how you make things happen, rather than just being zeroed in, like for me to say, well, it's all the mitochondria and nothing else.
Yeah, I think that's an important point. I know a couple of years ago, I had a conversation with Matthias Hebrock at UCSF, and I said, it's almost like it's a bad ballet. Like you have this beautiful ballet up on the stage, and then suddenly, like one of the ballerinas gets dropped, and then the next thing happens, and the next thing happens, and the next thing. It's like that. To me, it seems like, it's a sort of simplistic analogy, but it's sort of like the road to destruction of the presentation. So yeah, and I think that it is very important to understand that each piece has a role. So with that in mind, I wondered if, Dr. Wagner, Thompson, and Gaulton, could you each pinpoint and share your impressions of sort of unsolved problems that remain in this area of research?
Sure, I can go first.
Sure.
And I think that a lot of it has to do with things that we've just discussed in terms of cause and effect. We think about defective pro-hormone processing, for example. Is that a biomarker of what's happening, or is it a true cause of what's happening? My background, I'm a chemical biologist, so I like to think about things from kind of more on the therapeutics end of things. A lot of this reminds me of what's happened in Alzheimer's disease. So you have beta amyloid processing, plaque formation. For 20, 30 years, it's been thought that's the cause. And most recently, and we've all seen this in the news, the antibodies that Biogen has developed, they do attack those plaques and those beta amyloid processes, but they don't have an effect on the disease. And so thinking about, in our case, well, let's get to that answer more quickly than maybe that field has. And, I'm biased. I think about small molecules, and I think this is a way that we can start to tackle those questions. But to me, that's one of the major questions is, biomarker versus cause. Peter, go ahead.
Right. Yeah, I think everything, I agree with a lot of what's already been said, in the sense that we really we don't really know what we're looking at. I like that comment you made earlier, Amelia, about that. The tools that we're using, how good are they really at telling us what's going on? We're always going to be limited by that to some extent, but trying to be able to sort out things like cause and consequence, like you mentioned, Bridget, is going to really rely heavily on the tools we have available, and really, definitions. I think definitions are really critical in a discussion like this, because when we say things like pro-hormone dysfunction or mitochondrial dysfunction, it's a very vague term, right? And it's a very broad term. It can encompass a whole spectrum of of defects, in things like lysosomes, or in electron transport chain, or the processing enzymes and chaperones and inhibitors. So I think that all of those things are involved to some extent, but part of our task, I think, over the next while, and one of the major questions that I've always thought is, how do we start to parse out some of those things? Is it reasonable for us to expect that we can sort of isolate these processors, or look at them in isolation? I mean, Scott, you mentioned, we're all looking at this integrated system, so it's very difficult for us to kind of tease one thing out and say this is the thing that's driving a lot of the other problems. So I guess that's a lot of stuff to think about there. But really briefly, I just also want to add that when we bring it back to the human disease, and we're looking at this in the context of the natural history of type 1, we really want to know when this is happening. We need to know if it's something that's very, very early, in most cases, or or it's something that kind of happens later. And I think that some of the tools we have available, some of the specimens that we can collect now will help us to address that. So I'm really interested to kind of discuss this more and see where people's thoughts are with this, and also the relationship with endotypes. That's another whole nother discussion, but I'll leave it there.
Yeah, the temporal snapshot of what's happening as people progress towards presentation of type 1 is really an interesting place to talk about. Dr. Atkinson, did you want to sort of like weigh in here?
Yes, I would. In that, again, having been on the one of the people that was for many years just looking at the autoimmune side, there we had autoantibodies, we had a little bit of HLA susceptibility, and then T cells, which are complex. But if I now switch to this group's expertise and just what you've just said, the number of potential contributions seem to go up logarithmically, because each of you have your own biases. You've sort of admitted that. So my question is has the community of beta cell biologists sort of thought through how do you approach this when there's so many potential variables, and then not just approach it to segregate them, but how do you rank them? In other words, I think you're taking research to a whole new level here. So can you talk about some of the barriers you see, or how you think the field should move forward with respect to this area? That's kind of an open-ended question, but I'm really curious to hear your answers.
Can I maybe address that a little bit? I think that you're completely right. One thing that I think always bothers me is that we don't really know what normal is. And so, I think that there's a spectrum of normal. And until we truly understand that, I think it's going to be very difficult to then kind of assign a culprit to not normal and disease.
Yeah, you have to set your control group. What do other people think on that?
I think that's the Mark is asking the $1 million question. So I think the key is how do you solve an infinitely complex disease with multiple cell types that are all coming together, right? It's like a dysfunctional symphony, right? So to some degree, it would be lovely if there was one singular lab that could do that. But it's kind of like a behemoth of a problem. And so I think the first thing is to appreciate that, and then realize in order to get your hands around it, it's going to require complementary expertise. Absolutely. In many areas. And I think I love when I read papers where someone is distilling a whole disease into like 7 figures in a Nature or Cell paper. But like, let's be honest, that's not real. Okay. And so I think if we appreciate that there are some concrete pieces, and then we can dissect apart some of the concrete pieces, then we can start fitting together pieces of the puzzle. And so that's kind of how I've approached it. It's like when I see a patient, which is a very small percentage of my time now, but when I do, and someone is ill, we're trying to, look at the big picture, but realize that we have to start to zero in on something that we think is causing a problem, but not forget about everything else. And I think it's the same thing here. Like, we have to think about, look, we've got maybe 60 susceptibility genes. Those probably matter, right? We've got 4, 5, maybe more organ systems. Those probably matter. But now can we parse small pieces out and then tease those apart, whether it's looking at lysosomal function in Amelia's lab, or whether it's in Peter's lab, looking at how some of these DNA damage responses are turning on senescence, right? So, and then I think that what Bridget is doing is thinking big, because you're coming and attacking small molecules that hit multiple pathways, but then I don't need to speak for her, then you zero in on the hits, right? So I think we have to appreciate the complexity and then utilize our expertise to try to piece those bring those puzzle pieces together to get our arms around things.
And work together.
Yeah.
Yeah,
Of course.
I mean, I think that's key, and I couldn't agree more with what you're saying, and I think that, you know, I don't know if it's NIH's role or someone JDRF, but providing more of these kind of opportunities to work together. Right now, the single grant or even, you know, multi-PI, it's still very narrow scope and doesn't address exactly what you're saying, bringing in so much expertise. We're starting to get there a little bit in genetics, it seems like, with type 2 especially, like big functional genomics consortia to work out, you know, what are the effector transcripts and whatnot, blah, blah, blah. But I think in terms of physiology and biology, it's a little bit behind, and somewhere where I think we need to focus our attention.
I would agree. I think that that's why we decided to sort of launch this project, is because it's a multifactorial disease and so complex. There so many different disciplines look at it and study it, and so it has the siloed features of that, right? So this is a little attempt to sort of chip away at that and maybe sort of spark something different. But I would I'd also, just before we dive into a poll to the audience, you know, how do you guys think that, you know, the in silico solutions, you know, omics based approaches, or even AI that some of the, you know, companies in silico medicine and immune AI are now attempting, how do you think those strategies impact your specific field of inquiry? Anybody at all?
So Monica, while they're thinking of an answer, oh, I'm sorry, I don't want to.
No, please go ahead, Mark.
Let me just respond that I agree. I think there's a fundamental flaw in the system, and Marjorie's noted on this. And I'm not saying the typical route is not been productive, but I'm become over the years convinced more and more that self-assembling groups are better than grant competitions where somebody puts out an RFA and then you rank these proposals, and then the top scoring ones will get funding, and then they say go work together. I'm so much more convinced that if we switch the models up, where if you took people like on this call and said, hey, we're going to dive into this problem and we're going to dive deep, solve the questions together, and then go in with the proposal, that would dramatically accelerate progress, rather than this contrived system that's been in existence forever, where you say, okay, go work together. And that just instills the fires of promotion and tenure, publications, authorship, blah, 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 that we're all aware of, that are realities. And I'm not saying we can ignore them, but I really like that comment. I think there's a better way for the future. I don't know how to implement it, but I think we could do better. I just wanted to acknowledge that at a personal level, and that's just opinion. Others may disagree, but that's an opinion.
Yeah, I mean, I think you're saying go organic, right? It's very timely. Grassroots and organic. Start it up. I did want to just before we answer the in silico question, I have a comment from Frances Ashcroft. I think you all know who she is. She's coming over from England. I think there's a problem with our models. None are perfect. Beta cell lines are proliferative, and so their metabolism will not be identical to normal beta cells. They're often cultured at too high glucose. I've heard that many times. Min6 cells are cultured at 25 millimolar glucose, INS1 at 11 millimolar glucose. When islets are isolated, they're often cultured overnight in the hope of them recovering from the isolation procedure, but there's a problem because they have no perfusion, so the center of the islets quickly goes hypoxic. So metabolic is affected, and then there's again the problem of what glucose level to culture them at. We need to recognize this. And she said, I'm a beta cell biologist who focuses on T2 and neonatal diabetes. And we were talking about this a lot recently with Chris Hughes and his group. They've started a company, Arcadia, that is out of San Diego, and they're kind of working on these issues. They're trying to address them.
I guess I'll jump in and say something about that, because you know, if Fran Ashcroft is saying a point, I'm not going to be one to say she's wrong. But I think she's got her point is dead on accurate. I think we all contend with this problem. And so I think, you know, there's always issues with models. I think Amelia said that at the beginning. So there's two ways to look at it. Either one, we say our models all stink and we're not studying anything relevant and we take our ball and go home. Or we say, look, we have to make the best of what we have, attempt to build something better, but we certainly can't like give up, right? So I think, you know, nowadays it is very hard to publish a study with working just in a min6 cell line, because I think every reviewer is like, that's a problem. I think everyone realizes doing studies on ex vivo islets is also a problem. So what can we do? We have to utilize complementary approaches and then take into account the flaws in our systems at the beginning, but also realize if something is real, if something is critical, then by building multiple complementary pieces of evidence in parallel systems, including in vivo, I think that's the way forward. Obviously, we're not going to be, you know, doing a lot of in vivo experimentation in humans. So, you know, we have to realize that too, and do the best we can. Now, maybe, you know, some of the folks up in you know, Bridget's neck of the woods, they'll be building a perfect humanized model. And so then we can do that next. But I think it's hard. And then I think one other thing I would just generally say is it is very hard to just generally study a human, especially in type 2, but also in type 1. When we study human diseases, they take years to develop. And I don't know about any of you, but like my lab can't wait like 25, 30 years for a mouse to get died. Well, the mouse couldn't wait, right? But like we can't study things in the proper time context. So this is the part that stinks. And so we have to make the most of it. But I agree with Dr. Ashcroft. She's right on the money. Like these things aren't great, but we have to kind of recognize that and see if we can do better.
I agree with your point of using complementary systems. And I think this also comes back to your question, Monica, about the omics, that, you know, I think that they can be a very powerful tool as long as they don't exist in isolation, right? So they have to be complemented with other approaches to validate and whatnot. You know, I will say that I like the unbiased nature of a lot of them, those approaches, because you know, we're people and we're biased and we tend to look for things that we want to see. And so by taking that away, I think you can really discover important things. But again, you know, has to be complemented with other approaches.
Yeah, and I think an important point to that as well is having the well-crafted biological question. You know, I see all too often people use omics and AI, especially so trendy, as the tool that's going to give you the answer. And it's not, of course. You know, you have to have the right question and not just use it as a crutch.
Yeah, I would agree with that totally. I mean, I think that these tools could be almost like, you know, they could create a sort of a rich forest where scientists can kind of come in, you know, pick up pieces of the forest that they need and build a hypothesis from it, right? That's testable, and that's what they offer. I think that's my impression, sort of a basic impression. But
would everybody agree? Oh, I'm sorry. I know you got to get to your poll question, Monica. I'm so
sorry. No, go. Just let's go.
No. But would everybody agree that in this COVID era, despite it, this AI has become such a buzzword that everybody's talking about it. But, you know, my fear is it's just used as a buzzword. And I'm not sure we as a community are really approaching it well. And if you look at some of the AI-like efforts in type 1 so far, and I'm not calling out IBM here if anybody is on the call, but its help has been with Watson has been limited. Do people agree that we need to really, if we're going to go into this AI era, which I think we are, that we need to be careful about how we do it and really develop a good plan? Or should we just let it free flow and see what emanates?
No, totally agree with you. And I heard a talk pre-COVID, somewhat cynical statement, but especially here in Kendall Square, that you could just go onto a street corner and yell out AI or machine learning, and you'd have like 3 or 4 VCs like running up to you because they'd be like, oh, how can we do that?
No.
So, I view it with a pretty salty eye, and I think it's something where we have to just be really careful. I'm old enough now to remember back when, DNA microarrays were the big thing. And they were being developed in the late 90s, early 2000s. And it was a little bit the same thing. Oh, we have sort of a pseudo problem. Let's throw it on a microarray and we'll get the answer. And you just have to be careful. Yeah.
I think it's important to use the technology, but to use it wisely.
Yeah. Otherwise, we're just going to proliferate a lot of junk in the literature.
Yeah.
What's the old saying in the old I think it's a computer modeling or computer biology saying, garbage in, garbage out. Right?
Exactly.
So I mean, if you're building these AI models on like trash, what do you expect that you're going to get at the end? So I still I don't want to completely disregard them either. So I would only say this to you, Mark, that if we're going to solve an exceedingly complex disease, we need to utilize all the tools. It has to be an all of the above strategy. And I think everyone is right. We need to be reserved in how we apply every single individual tool. But I also am not like totally ready to say like, this is all garbage.
No, absolutely. Right? And Dr. Ashcroft put a great comment in the chat about AlphaFolds, which just came out. And that I would agree, that is amazing. So, there's a good role for these complex.
But I don't think any structural biologist would say I'm out of a job now because of that, right? So it's a piece of the puzzle. Exactly. Yep.
Precisely.
Again, I just would say from my sort of a long view or, sort of a basic view, it looks like these are all new tools that are there to be used to develop hypotheses. And then you really have to go through the rigor of testing them, right? Am I right there? So that's kind of, that's an impression. But I'd love to ask this poll to the audience. I'm going to put it out here. Just to sort of circle back to everyone's work. And the poll is, what is your best guess as to the link between mitochondrial dysfunction and pro-hormone dysfunction in beta cells? I'm going to launch it and I'm going to let the panelists vote. So everything's anonymous. Basically, it's, ER stress, glucokinase downregulation, lipotoxicity, underlying mitochondrial genetics, or other. And there was a very interesting paper, the MOTS-C paper that came out, I think, yesterday, 2 days ago. A USC group and a Chinese group put that out there. It's kind of interesting about, that points to some mitochondrial genetics. But of course, it's almost like there should be all of the above as well as one of these. Well, it looks like ER stress is in the lead at 75%.
Is that a surprise?
No, I mean, there's, of course.
I feel like when you put the poll together, I knew that was going to be the trendy answer.
Okay, well, let's dissect this. What do you think, Dr. Thompson? What is your thoughts on this?
Yeah, I mean, this is, it goes back to what we were saying earlier. I mean, all of these things we know have a role to play. So, it could just be a timing thing, like, at certain stages in the pathogenesis. Again, going back to the natural history, we want to understand how this thing unfolds, and especially looking at the context of, different endotypes. There's growing evidence that we cannot just treat type 1 as a one size fits all. It's the same thing every time. People who get it younger versus people who get it older, there's a difference there. So, the question is, maybe it's ER stress for people who get it later, and then maybe it's something like mitochondrial genetics, and I would be partial to say DNA damage and senescence, or other things that could also play a role, depending on the endotype. So I think it's a really contextual type of question, and we have to really understand or think about the context in which these things could be happening.
Yeah, that's good. That's a good summary for sure. of course, there's the whole viral insult, Coxsackie, and of course, that's a big one to drive the ER stress. And a lot of people are really examining that and counting on that as a way, a workaround for preventing type 1 is to get rid of the Coxsackie infection in the first place, right? So,
but I guess the question is, and this kind of gets at Peter's point, is that, will addressing that one thing solve all the problems? And the answer is probably not. I think that taking this kind of very focused one track approach to it is perhaps going to help some people, but broadly, it's not going to help everybody.
Yeah, it'll be interesting to see, I guess, if there was this whole push for Coxsackie vaccinations, then what happens to the population? Do we see a decrease in T1D or insert some age-related populations or what? I guess, let's see if there's a bunch of questions.
Can you just quickly cover this ER stress thing?
Yeah.
Because I think it's important to stimulate discussion. I'm not trying to like trash on ER stress. I mean, my closest collaborators here at Michigan all study the ER. Some of them are on the call. And clearly mitochondria intersect with the ER quite a bit. And we study the mitochondria extensively in my lab. But I feel like we have paid a lot of attention to the ER in both type 1 and type 2, which is fine. I mean, I think it's not that it's not important, but I almost feel like we've paid attention to it at the expense of thinking about other stress response pathways.
Mm-hmm.
I mean, we know very, very little, and again, I'm going to call people out, but we know very little about regulation of autophagy and beta cells. We know very little. We know even less about regulation of senescence, which is good for Peter and Amelia because they have lots to work on.
Yeah.
We know a lot about ER stress, but I feel like we have an exceeding focus on this as the catch-all that is the root cause of type 1 and type 2. And I'm not saying that I don't think ER stress is important by any means. All I'm saying is I feel like it is the catch-all for all problems in the disease. And even if you look at the topic of discussion, it's like mito dysfunction and pro-hormone. It's like, oh, well, what regulates pro-hormone conversion? Oh, well, a lot of insulin processing, starts in the ER. So of course we're going to talk about ER stress. So I want to, maybe I'm being a little adversarial. That's not my goal, but I feel like we need to talk about, like, are we all just a bunch of ER biologists? Is that what the beta cell field is? Because I feel like that's sometimes where we often go with every topic. It's like, how did this thing lead to ER stress to kill our beta cells?
Yeah.
Right.
This is great.
I don't know.
So Scott,
is it
Great.
fair
I
to think
love
about
that
things?
comment.
Great.
So what you're saying is, if in a field that's already hard to get funding for, and again, that's the fuel for our lab, there's a Pied Piper effect in a way, and that we're all working toward the same thing, which may be involved, but we need more diversification in thought. That's what you're saying, right?
Yeah, I mean, essentially, I think that we seem to be a little bit over focused on it, and I think that that's at the expense of other topics. And I think that, again, if it was it's different. Like if you're, if you're Fumi Arano and you're studying, Wolfram syndrome, monogenic disease, like it's very clear that's got to be the focus. The ER is really important. I don't have any question about that. Type 1, not a monogenic disease. There are many genes. A lot of them have nothing to do with the ER.
Mm-hmm.
So it's not that I don't think ER stress is critical. It is, but I think it's like one piece, and I feel like we don't think about the other pieces enough.
Well, on that note, I mean, do you want to, Drs. Linneman and Thompson, you want to dive in a little bit on autophagy and senescence and why that's important to appreciate?
Well, so I'll start by saying that Peter and I keep talking about discussing this further together, and we haven't done that yet. So, so this, it's a little bit premature of a discussion, but I think, it kind of goes back in with the chicken versus egg question. What's the cause? What's the consequence? When does ER stress come into play? And is it, a problem because of things that happened before due to genetic susceptibility, combined with a specific environmental stressor that, in that set of beta cells, you have this particular response? Peter, I don't know, do you want to add to that?
Yeah, I think it is a complex issue. I think different stress responses can involve other features that, you might not necessarily think that they, co-occur together. So for instance, autophagy and senescence, I think there's good evidence to suggest that, autophagy, defects in autophagy could be contributing to senescence, or, autophagic processing is a component of senescence, for instance, right? But, we're not thinking really on that level. And I think it is because, as Scott mentioned, there has been a unilateral focus on ER stress, unfolded protein response. This has dominated the idea of what happens in the beta cell in type 1 diabetes.
Yeah.
And there's good that's come of this, because we've seen clinical trials. This field has really progressed quite a lot from, initial studies in NOD mice through to validation in humans, and some clinical trials that have come out. One with verapamil, the TXNP inhibitor that Aneth Shalev's group has published a few years ago now, and showed some effect in recent onset type 1 patients. And then very recently, Steve Gittleman's group at UCSF published a really nice study in Lancet on the use of imatinib as a way to spare beta cells. And this was based on a large body of earlier work showing protective effects in type 1. And this is also targeting components of UPR and ER stress. So, we look at this, we think like, oh, wow, we've really just focused on this main thing, and everything does seem to kind of feed into that. But we have seen some fruit from that too. And I think that it's important to balance that out. So I do agree with you, Scott. I'm not trying to jump on the other side of the.
Oh, no, I'm not. And I'm not, I wanted to, I brought this up more in let's talk about it, rather than I think that ER stress is not important at all.
For sure, yeah.
I actually do think that there's really good literature. What I'm worried about is that becoming the only literature?
Yeah, absolutely.
Right. And that's what I'm saying. And I think Mark brought it up too. When you're Pied Piper, you're writing grants. It's well, if I don't mention ER stress, then I'm not going to get funded, right? I'm worried about that, because I don't think all of the signs point to that. I think there are signs for sure, but I think we have to think a little bit more broadly.
Yeah.
I mean, and maybe I'll pester Bridget to tell me, when you're doing, pharmacologic cellular targeting, is it all about ER drugs, or I'm assuming there are a lot of other really interesting targets for beta cells to make them function better.
And I think we've had a lot of success kind of taking it more from the phenotypic approach. We just want to, by hook or by crook, allow beta cells to survive in the presence of some insult, for example, and then work our way back to figure out, well, what did those compounds do to protect the beta cells? Very frequently, it's not ER stress, and it's some other target we would never have guessed otherwise. And so that's one area where, opens up a lot of biology for us to pursue, which is great. But yeah, I think about it in terms of, are there, as you mentioned, Scott, is everything funneling into ER stress, multiple insults funneling in? You could imagine another model where one insult causes many different types of dysfunction, or is it all very parallel? And so the kind of everything bagel where we're going to be in trouble.
Well, and just to kind of add on to that, if we're so focused on ER stress and we're targeting ER stress, are we missing something key that is upstream of that? And, like you said, Bridget, if it's funneling into ER stress, is solving that problem really going to solve the problem?
That's right. Exactly right. And I think part of the problem with ER stress is, like autophagy, you need a balance. And so a compound that just wipes out ER stress is not going to work because you need a little bit of ER stress, right? So it's really tough.
It's extremely complex.
But I'll say this, the one avenue open, and sorry, Monica, to keep you from your poll questions, but there is
good.
therapeutic opportunities here in this space. In that, again, if you look at we just put a review on this, whatever it is, 100 therapeutic trials that have occurred in the last 12 years in type 1 diabetes. Almost everyone has been in immune therapy. So I'm not discounting your collective works, because it could be that, your efforts are going to lead to new pathways of trying to target not just the immune response, but things at the beta cell level, and perhaps therapies that have been developed in the type 2 diabetes world, if they can be rendered specific enough, might form something additive to type 1 intervention efforts. But the field of immunologists as a whole are really having a hard time grasping, and I'm calling my own, I'll only point to myself here, but they're having a hard time grasping everything that we've talked about the last 45 minutes.
Oh,
well, Mark, can I ask you about this, since we're talking about mitochondria, and your colleague and my dear friend Clay Matthews is one of the folks that's been leading the charge on mitochondrial biology in the immune system. I mean, are any of those, I don't follow the immunologic trials, but I feel like there are any of them targeting immunometabolism in type 1 right now? Because I feel like the literature on metabolic switching in the immune system is, at least in the preclinical models, pretty compelling, but I don't know if that's reaching the bedside or not. And maybe it should, because again, Clay's data over the years and the genetics on that has been pretty interesting.
Right. And again, with a dose of humility, the immune therapy side has been just essentially target bombing anything in the immune system and seeing if it sticks, meaning a lot of trials. And it's not for unjust reason, but all those commercials you see on TV at night where it says this drug prevents plaque psoriasis, or this drug is effective in RA or whatever. If it's safe and you can find something in the literature that would point to, well, this may impact type 1 diabetes, this has driven many, not all, but many immune intervention trials in type 1. And that's only got us so far. And, with the recent Provention, one of the sponsors here, we are ever so close to seeing the first approval of a drug to delay type 1 diabetes. But again, I think the research line that you all are looking at forms a real effort for the future in terms of making things a lot better.
Yeah. No, that's really true. I mean, if you have to for real progress to occur, I think you have to look at the good and bad points. What needs to be done, and not just what's being done. And the audience has done this today, I think, to really uncover some places for improvement, which is, of course, that's sort of what a scientist looks for. Like, where can I go? What can I do? What experiments can I do? I just want to go, because we're closing in on our time, but I'm going to skip the second poll and go to poll 3. And this poll is really kind of granular. So it's interesting. It's, sort of coming out of, it's Adam Ramsey, who's an, I believe, MD PhD, up with Tim Kiefer up in Canada. And they just had this a preprint come out, and basically the question for the poll is it still valid to assume that PC2 exists in the beta cell, given recent somewhat controversial literature suggesting there's no PC2 in the beta cells, but PC1 third is the only pro-hormone convertase, and that's Ramsey et al., Diabetes 2020, July. So looks like there's a lot of maybes out there. 83% maybe, no one's committing. Okay. All right, so I'll end that poll. Looks like maybe. Who's interested in commenting, taking that bomb?
I'll start here,
Okay.
just because I'm such a huge proponent of really trying to study things in the most physiologic way possible. I mean, we do a lot of work with intravital microscopy because I like, not disturbing the system until we're looking at it. But I think it depends on how you study it, what you're going to see, first of all. Second of all, it's all about context. When are you looking? When do these things change? Do they change?
Mhm.
I think there's a lot of stuff we don't necessarily know yet. I don't know if anyone else wants to add into that. No? I mean, I think that perhaps in certain systems, you may not have high expression of that, right? I mean, I think it's very clear. But what about other scenarios?
Yeah.
So I was a maybe on there.
More
Yeah.
more work needs to be done in that system. I mean, right? That's what it seems like. I'd also sort of like, before we do another poll, I want to ask people, this paper came out the other day, Byung-Soo Kong and co. basically came out, mitochondrial encoded MOTS-C prevents pancreatic islet destruction in autoimmune diseases. Whew, that's a big one, right? I mean, autoimmune T1D can be prevented by MOTS-C peptide treatment in NOD mice. And we know that MOTS-C is a 16 amino acid peptide. It promotes, sort of metabolic balance. It's mitochondrial, peptide. So that's weird. What do you think of this?
Scott, I vote you start. You're
Oh,
the
God.
token mitochondria guy.
I'm gonna
Yeah.
put in the chat.
I was hoping Bridget would go back and call on her days with Vamsi Mutha and be able to tell me the answer.
No.
She's like, no. I've looked at that study. I mean, this builds on the original identification of this MOTS-C, this small ORF that's derived from non-coding regions of the mitoDNA. That was published by, I think, Cohen and the like in maybe 2015.
2015.
Interesting,
Exactly
interesting
right.
original work on that. That peptide, in that original cell metabolism paper actually improved glucose uptake by peripheral muscle cells and improved body glucose control and body weight and insulin sensitivity in the muscle. So now this is the interesting thing in that study was that they suggested that glucose uptake and glycolysis was enhanced by MOTS-C. And then this study, they say, actually, no, this causes T cells to metabolically switch from glycolysis to oxphos and then makes these T cells more, less, diabetogenic, if you want to say it as such. So my answer is, I don't know, because I think these are two isolated studies that are showing very distinct roles for this peptide in very different cell types. I love the mitochondria, but I'm also not magic bullet guy. So, my initial answer would be, I feel like Mark might have written this paper like maybe 20 years ago of like the gigantic list you can't even fit on one page of things that cures type 1 in an NOD mouse. Was Mark, that was you, right? So like, I just am like, great, okay, it worked. That's cool. But this peptide also did completely different things metabolically in a completely different organ system. So I just don't know what to do with it other than say it's cool, but there's more work to be done.
Yeah, that publication was a classic example of what not to do to win friends and influence others. That's all I can say. You don't kill the goose that lays the golden eggs. So anyway,
I would argue, though,
I appreciate those
things,
comments.
sorry, Mark, to interrupt. I was just going to say, I would argue that things worked out okay for you.
Yeah. I know
It did, and thank you. But let me put one question to the group. I'm loving this call, by the way. So you put the chicken and egg notion out there earlier of immunity versus beta cell defects. What if we were going to experimentally try to find the primordial sin here? And that may not be the appropriate word, but it's the one on my tongue right now. In terms of the beta cell, how would you prioritize these, or how would you explore that to see what define the pathway, like this occurs first, then this, because it seems like we're in a love fest here for beta cells being involved in their own demise. But how are we going to approach that question?
Perfect.
And hopefully it's clear.
That's perfect challenge.
Maybe I can start it off by saying that, without getting into any one specific pathway or model system, I think the way forward is going to be, I don't know who said this, Bridget or Scott, maybe, about taking this approach where none of us are experts in everything, so therefore we all have to work together to contribute our expertise to these maybe umbrella studies that are going to move the field forward. I think a part of that is again, I'm biased about model systems, but I think that a key way forward is going to be to continue to work on developing the way we study type 1 diabetes, type 2 diabetes, the way we study the beta cell and the immune system and how they interface. Because, crap in is crap out, as Scott said. And so if you, if you want to get the answer, you have to come at it in a number of different ways. You have to develop the appropriate systems to be able to do it effectively.
Yeah, that's well said. Collaboration is key. I mean, Ekaterine Bershvili said it. The time of the individual researcher in type 1 diabetes is over, and collaboration is so necessary, particularly in a multifactorial disease and a layered disease, in a time frame driven disease. It's like everyone has to contribute their piece, I think, to really build a true model of what's actually happening. Mark, did you want to say anything more to that point?
No, I'm in agreement. Like I said, to me, this is a love fest. I just want to hear more from the true experts here. I mean, Amelia laid a great case. Who would add on to that?
I'm certainly no immunologist, but it seems that the immune system recognizes something about the beta cell. And the question becomes, did the immune system pick that up first, or did the beta cell die first and start to release antigens that the immune system is recognizing? So figuring out maybe that order would maybe get at Mark, at your question about, is the beta cell responsible for its demise or more of a victim? Yeah.
From the audience, someone asked, this is just sort of a topper, like if you were given unlimited funding in human resources, where would you focus your work in the context of this problem we're discussing? What group, I mean, we're talking about consortia and gathering scientists, like who would you have in the room? What experts would you want to have in the room to build a better appreciation of what's happening?
Can I want to answer your question, Monica, and then Mark's. Maybe I'll do it at the same time. So I think the one thing I really want to, I'm glad that we love the beta cell in type 1, because we did not love it for a long time. But I think, again, I will make the point. You have multiple organs that are all sick. I am not a believer that it's only the beta cell causing the problem. I'm also not a believer that it's the immune system causing the entire problem. And so we need to, it has to be an all of the above approach. We need to study the exocrine pancreas, the beta cell, the immune system, and the microbiome. And there are probably other systems involved. And I think if you think about it as a dysfunctional symphony, rather than let's find the one problem child, we'll end up sniping. I feel like we've spent way too much time. I've read all of this homicide, suicide papers over and over, and I'm like, we're all wasting our time. We need to recognize there are multiple problems and get together and study them all in parallel. I think that's a better approach. And then in terms of, like, if we had unlimited resources, so if I had the unlimited resources, my goal would be to use my unlimited resources to give unlimited resources to my colleagues. Because again, it can't be one thing. It's got to be everybody. And so that, maybe somehow I can use my unlimited resources to get Jeff Bezos and Bill Gates to pay for all of us to take care of this as a group.
Let's get them. Chan Zuckerberg, come on, help. Let's help the diabetes research community sort of circle the wagons. I would say also to that point that just to sort of to end it up, that you know, there's a wealth of really talented scientists out there. This is a very complex problem. It's a really exciting problem, and I think it's really at a tipping point in many ways. Just in terms of scientists, young scientists entering the fray with new tools and sort of the mentorship from the seasoned scientists. There's a lot of good things that are there. And I think I don't want to say, well, I'll say it anyways. Type 1 diabetes could be a poster child basically for how to get things done in a multifactorial disease. And I think that's starting to kind of coalesce, and I'm really happy to be witness to it. And I would say again, thank you all so much for joining and talking and discussing and being very honest about, you know, things. All of your labs are really esteemed and amazing, and I think that young scientists listening to this will, you know, be like, wow, that sounds really interesting. Maybe I want to like call, see if any postdocs are available, or, you know, positions. So I hope that also happens. I would say, you know, our next town hall style State of the Science is next week, August 5th, prevention and T1D, remission or cure. We have a bunch of sort of biotech startup people involved there, and that will be interesting too, sort of bringing things to the clinic, and how that all is progressing. And, you know, again, this whole exchange will be available recorded on our YouTube site. So thank you again so much for everyone's time and for all that you're doing in the space. You're absolute heroes.
Thank you.
Thank you.
Thanks for the opportunity.
Thank you. Yeah, absolutely.
Thank you again.
Bye, everybody.
Bye-bye.
Everybody.